Hello, boys and girls. I'm glad to be back with you guys. I hope you guys are safe, are well rested, and are ready to start learning again. I will apologize for some of the noises that are happening outside. I'm here in the porch of my house. We're going to hear a lot of cars and a lot of kids also who are playing outside. But let's just try to ignore them and move on with your lesson. Okay, so for this lesson, we're actually going to go into a very specific topic that's affecting us during this whole quarantine thing, which is the coronavirus. But before actually going and talking about the coronavirus and what it does or what it is, I gotta teach you guys first of all what is a virus in general. Okay, so with that in mind, we need to start over here. First of all, what is a virus? A virus is a tiny non-living particle that enters and reproduces. That term is actually incorrect. A virus does not reproduce itself. It replicates. I'm going to explain that in a couple of minutes. Reproduces itself inside a living cell. Virus particles are tiny compared to bacteria. If we look over here, these here are examples of different types of viruses. You have the smallpox virus, which is the biggest one in this example, and then you have this one over here, the yellow fever virus, which is the smallest one in this example. This over here is a Streptococcus bacterium. It's a bacteria. And this red dot over here is a red blood cell. So as you guys can see, viruses are extremely, extremely small. So then, living or non-living? Are viruses living or non-living things? Well, if we go back to your original lesson during the year, it says in here that viruses don't follow the characteristics of living things. Okay, so they are considered non-living. The characteristics of life, if you remember them from your lessons, are orderly structure, which viruses in this case do have. Produce offsprings. That means that they reproduce. No, viruses do not produce offsprings. They are incapable of doing so alone, which is why they need a host. They need a cell or a bacteria to actually help them replicate. But by themselves, they cannot do it, which is why it is said that viruses cannot reproduce, grow, and develop. Viruses don't grow and they don't develop, okay? They simply replicate themselves. One virus will produce 1,000,000 copies of itself, but these copies will not grow and develop. They might mutate, but that's a whole different topic in itself. And they adjust to changes in the environment. They react to them, but they are unable to adjust to them, okay? So, because viruses don't follow these three characteristics over here, they are considered non-living. And one more thing, viruses are not made of cells. So, in your book, and as well if you do some research, you're going to know that there are 4 basic types of viral shapes. But we're going to focus on these 2 over here. The first one is this spherical shape, okay, which is the shape that the coronavirus has. And now we're going to focus on this one over here, which is the T4 bacteriophage. Let me move myself a little bit over here, and that way you guys can see. So in here you have the T4 bacteriophage. Alright, so we're going to focus on these 2 shapes of viruses. Okay, so a virus. What is its structure? What is it made of then? If we start here at the very center of the virus, viruses can be made of 2 things at the very core: DNA or RNA. We're going to talk about both types separately later on. This RNA or DNA is enclosed inside a membrane, which is called a capsid. Alright? Both of them together are basically the core, the nucleus of the virus. Okay? This nucleus, this core, is actually enclosed in another membrane over here, which is called the tegument. And finally, all of these layers are covered by another layer, okay, which is called the envelope. The envelope is kind of like the cell membrane, but it's quite different in many things. And finally, we have in here what we call glycoproteins. I need you guys to keep this in mind because these are really important for later on during this lesson. So these are like the main parts of a virus, okay? There are many more, but these are the ones that you guys need to keep in mind for now, especially the glycoproteins, these little like arms that extend outside of the virus. Okay, let me move myself over here, maybe over here, there we go. Aha, so then, in here we have viral replication. For actual viral replication, I need you guys to go back to your phospholipid bilayer lesson. If you remember, among other things, the phospholipid bilayer is made out of these proteins over here, okay, and this carbohydrate chains, all right, and just like from the virus, they are also called glycoproteins. So the combination of this protein and this carbohydrate are called glycoproteins. But it's quite interesting, right, that they both have this glycoproteins. So what are they useful for? Okay, in this next section, I'll explain why. Let me pause the video a little bit and prepare the next segment of this lesson. Welcome back, boys and girls, and here is the next part of the lesson then, the viral replication process. There are actually two different processes. The first one is called the lytic infection, and the second one is the lysogenic infection. Okay, so we're gonna first go into the lytic infection. Do you guys remember from the viral parts, the glycoproteins? Well, in this T4 bacteriophage, these ones are it, okay? And this is a bacteria, and bacteria also have glycoproteins. The thing is that certain types of viruses, can only attach themselves to a specific type of glycoprotein. In this case, if you notice in here, this virus has little round glycoproteins. In a way, they act like chains, and they allow that virus to actually infect the bacteria. So it grabs onto the glycoproteins of the bacteria or the cell, and then it simply opens like a door. Literally, the cell membrane opens up if the glycoproteins are actually like keys, or they actually fit into each other. So then, here the virus will inject its DNA or its RNA into the cell, and this nucleic acid is gonna take over the cell. It's gonna take over the protein synthesis process. It's going to start making copies of itself. So as you can see in here, as it makes copies of itself, these copies rearrange into each other, and they will eventually burst, making a whole new bunch of viruses which are identical to the first one. Okay, so that's lytic infection. But what about a lysogenic infection? Look at the glycoproteins in this virus over here. They actually can only fit this bacteria over here. If I try to grab it and put it in this one, for example, which has triangular glycoproteins, it will not fit. So viruses know which glycoproteins to attack. Now then, same thing over here. The glycoproteins connect each other, and the bacteria or the cell membrane opens, and it allows the virus to inject its DNA or its RNA into the bacteria or the cell. But this was a little bit different. lysogenic infection actually takes time. And what happens is that the DNA becomes part of the DNA of the host. And it will only start replicating once the conditions are perfect. Have you ever wondered why is it that you get a cold sometimes, you start sneezing or whatever, and you don't know where you got it from? Oh, that's because you had a lysogenic infection. The virus was actually waiting for the right conditions to separate itself from the DNA of the host and immediately start its replication. It takes over the protein synthesis process of the bacteria or the cell, and it will eventually it will force the cell to make parts only for new viruses. And when the new viruses have been assembled and everything, they will burst out of the cell. Okay? So, in a nutshell, that is what a virus is. Okay? There are many more topics about this that I am not touching, that I'm not going into, but that is what you guys need to know for now.